Assalamu alaikum everyone, I'm Tuba nafees, presenter at SBA. Back to you with another topic, blood basics. It will cover blood cells formation, composition of the blood components and their function. Let's begin. As blood is composed of different types of cells, it's a fluid connective tissue. Approximately 8% of an adult's body weight is made up of blood. Females carry around 4 to 5 liters, while males carry around 5 to 6 liters of blood in their bodies. Its mean temperature is 37 degrees Celsius. It has a pH of 7.35 to 7.45, making it slightly basic. Whole blood is about 4.5 to 5.5 times as viscous as water. Blood in the arteries is brighter red than blood in the veins because of the higher levels of oxygen found in the arteries. Now move to composition of blood. Blood is mainly composed of plasma and form elements. Form elements constitute the solid portion of the blood, while plasma makes liquid portion of the blood. Plasma contains amino acids, nutrients, proteins like albumins, fibrinogen, globulins, electrolytes, as well as waste products to be discarded or discharged like nitrogenous waste products and toxic gases. Form elements consist of platelets, leukocytes or white blood corpuscles, and erythrocytes or red blood corpuscles. Leukocytes have further subdivision. They are divided into granulocytes and agranulocytes. Granulocytes, as their name suggests, are the types of white blood corpuscles or cells that contains granules. I will discuss the function of these granules and the classification of granulocytes in the upcoming slides. Granulocytes are again subdivided into neutrophils, basophils, and eosinophils. An easy way or easy mnemonic to remember these names is Ben 10 Game. It was an American series, I think. Ben means basophils, eosinophils, and neutrophils. And with the letter G, you can remember granulocytes. So it's easier to remember Ben 10 Game. Agranulocytes. These are the type of white blood corpuscles that lack granules, so they are called agranulocytes. They are further divided into lymphocytes and monocytes. AMLA is another mnemonic to remember the names of these white blood corpuscles. I hope it will be easier to remember now. Now move to blood components. When a sample of blood is spun in a centrifuge machine, the components get separated as plasma and form elements. You can see the distribution of the components like plasma, buffy coat, and erythrocytes. Plasma constitutes 55% of total blood. Erythrocytes or RBCs constitute 45% of total blood. And in between these two components, there lies a buffy coat that consists of leukocytes and platelets that forms less than 1% of total blood. Buffy coat is so called because buffy it refers to the buff that is brownish yellow color. And this is the situation when a blood sample containing anticoagulant is spun in a centrifuge. Because of the presence of anticoagulant, the clotting factors remain present in the plasma and fulfills the definition of plasma that I'm gonna discuss later. Plasma and serum, there is a major difference between these two that you need to understand. We often get confused between these two terms. Just remember the mnemonic PC, P for plasma and C for clotting factors. Keep in mind that plasma contains clotting factors. If a blood sample containing an anticoagulant is spun in a centrifuge, we get plasma. Because anticoagulant prevents clotting factors getting engaged in coagulation process, so the clotting factors remain present in the liquid portion of the blood or plasma. On the contrary, serum doesn't contain clotting factors, and in order to get serum, we spun a blood sample lacking an anticoagulant. Because of the absence of anticoagulants, the clotting factors remain non-present in the liquid portion of the blood because of their engagement with the coagulation process, and so we get the serum. RBCs or erythrocytes are also called hematoids, erythroid cells, or red blood corpuscles. They are non-nucleated. They lack a cell nucleus in most organelles in order to accommodate maximum space for hemoglobin that is responsible for the red color of RBCs and blood. In humans, mature RBCs are flexible and oval biconcave, so they can move easily through the blood vessels. On the inner surface of RBCs are two proteins called spectrin and actin that give the membrane resilience and durability, like they protect the cell against environmental stress. The plasma membrane of a mature RBC has glycoproteins and glycolipids that determines a person's blood type and makes the people blood unique by giving the specific blood group. The diameter of RBC is around 7.5 micrometer, and the thickness of RBC is approximately 2 micrometer. As I have already discussed, the hemoglobin present in RBCs is responsible to transport oxygen to the tissues everywhere in the body. This hemoglobin can also bind to carbon dioxide and becomes a molecule called carbaminohemoglobin, and in this way it can be discharged from the body by the process of expiration. Let's discuss erythropoiesis. Erythropoiesis is the production of erythrocytes or RBCs. RBCs are produced in bone marrow. They originate from a stem cell that goes through several stages of development. As you can see, pluripotent hematopoietic stem cell develops into proerythroblast, then proerythroblast develop into erythroblast. Erythroblast is the stage where ribosomes are synthesized and hemoglobin synthesis takes place. Next to erythroblast is reticulocyte. Reticulocyte is the last stage of RBC production in bone marrow. Reticulocyte spends 3 days in marrow and 1 day in blood. It releases its nucleus and develops into erythrocyte. As we have already discussed, red blood corpuscles are non-nucleated. Erythrocyte or RBCs have a lifespan of 120 days. As the red blood cells become older, they are further processed. They are phagocytized by macrophages in spleen, liver, or bone marrow. Their hemoglobin content is released. The iron in the hemoglobin is reutilized by the body, and the globulin portion is reutilized for protein synthesis. And that iron is again in bone marrow, but in infants and children, it may take place in spleen and bone marrow as well. You can see the multipotent hematopoietic stem cells have two lineages: lymphoid progenitor cells and myeloid progenitor cells. Lymphoid progenitor cells give rise to B cells, T cells, and natural killer cells, whereas myeloid progenitor cells are further classified into myeloblast that give rise to granulocytes and an agranulocyte, that is monocyte, whereas megakaryocyte give rise to platelets. Then we have reticulocyte that give rise to erythrocytes. And this is how the HSC give rise to different type of blood cells. Unlike RBCs, WBCs, called white blood corpuscles or leukocytes, are nucleated. You must remember, we must have heard the term police of the body for WBCs. They are so called because they protect the body against infectious diseases and foreign invaders. WBCs are further classified into granulocytes and agranulocytes. Let's discuss them one by one. Granulocytes. Their cytoplasm contain granules that are filled with enzymes that protect the body against invading pathogens. As I have already discussed, they are further classified into neutrophils, eosinophil, and basophil. They constitute 65% of leukocytes and are important in innate immunity. Because of the different shapes of nuclei, they are called polymorphonuclear. They originate from bone marrow. Agranulocytes don't have granules in their cytoplasm. They're classified into lymphocytes, macrophages, and monocytes. They constitute 35% of leukocytes. They are important in adoptive or acquired immunity that occurs after exposure to an antigen from a pathogen or vaccination. Their nuclei don't have different lobes as granulocytes have, so they are mononuclear. They originate from lymphatic system or bone marrow. Let's discuss the subclassification of granulocytes in detail. Neutrophils, like other granulocytes, are polymorphonuclear. The nucleus of a neutrophil contains different lobes that vary from 5 to 7. Their cytoplasmic granules are very fine. They make up around 60% of the total granulocytes, and they are important in destruction of bacteria and the release of chemicals that kill or inhibit the growth of bacteria. Eosinophils. They have large granules as compared to neutrophils. The nucleus has two lobes that can be clearly seen under the microscope. They destroy allergens and release enzymes that disable parasites. Basophils. Basophils have a nucleus that is hidden by granules. These are present in the least amount and are found in bone marrow or bloodstream. They mediate allergic reaction and prevent blood clotting. So eosinophils and basophils are mainly involved in allergic reactions. But basophils are often seen in the blood smears under a microscope in case of different blood disorders. Lymphocytes. Lymphocytes are further classified into B lymphocytes and T lymphocytes. B lymphocytes, they make antibodies that can neutralize antigens on the microbe surfaces, and they are important in the reactions mediated by T lymphocytes. T lymphocytes are further classified into helper T cells. They release a protein called cytokines. This protein stimulates the responses of other white blood cells. Cytotoxic T cells, also known as natural killer T cells, as their name suggests, they kill cancerous cells, virus infected cells, and the cells that are damaged otherwise. Memory T cells, they will be produced after the body has been exposed to antigen or any infection and prevent the body against the further infection of the same type. Regulatory T cells, also known as suppressor T cells, help to regulate other T cells to prevent them targeting the body's own cells. Monocytes form another classification of agranulocytes. They are further subdivided into dendritic cells and macrophages. Dendritic cells are basically antigen presenting cells, as they present antigens to T lymphocytes that stimulate further immune response. Macrophages phagocytize the cells. They are phagocytic cells. Neutrophils do the same function, but there is a difference between these two that you need to understand. Neutrophils perform phagocytosis, but they don't present antigens to the T cells. Unlike macrophages are phagocytic cells, which are larger and live longer than neutrophils, and they present antigens to T cells as well. Moreover, you already know that there is a morphological difference between macrophages and neutrophils. Their neutrophils are polymorphonuclear, macrophages are mononuclear. Neutrophils are important in innate immunity. They appear first against the invading pathogens, whereas macrophages come later. So this is the basic difference. Platelets are small fragments of bone marrow cells, and therefore they are not really classified as cells themselves. They are the fragments of RBCs, but they have very important function. They secrete vasoconstrictors, which constrict blood vessels. The blood vessels become narrow, so the lesser blood is lost in this way. They form temporary platelet plaques to stop bleeding. They secrete procoagulants, clotting factors to promote blood clotting. They dissolve blood clots when they are no longer needed. They digest and destroy bacteria. They secrete chemicals that attract neutrophils and monocytes to the sites of inflammation, and they secrete growth factors to maintain the linings of blood vessels.